Hi, this is Diptikan Padiari, lecturer in botany. Welcome to my channel Mikan's Botany. Please subscribe my channel and hit the bell icon. Hi, in this session, it is the second part of the prokaryotic DNA replication. After helicase protein binding to the circular DNA, what happens? SSB protein will bind to the DNA strand. SSB protein means single stranded binding protein. SSB protein present in the terminal tetrameric form and binds to the single strand of DNA for its stabilization, because due to its binding property, two DNA strand can regain its original structure. So avoid to such kind of problem, SSB protein binds to the single strand of DNA and stabilize its single strand property, stabilize the single strand. Then the next protein will be DNA gyrase protein. It has the topoisomerase activity. The torsional strand, the due to the unwinding of DNA on both the direction, there will be a positive supercoiling on the behind the helicase enzyme. So this positive supercoiling is removed by the DNA gyrase enzyme. Okay, the torsional strain generated by the helicase action, that positive supercoiling, is relaxed by the DNA gyrase enzyme. This is a DNA gyrase enzyme is a topoisomerase 2 enzyme. It has the potential enzyme. It is a potential enzyme against anti-cancer drug. Sorry, potential enzyme for cancer treatment. The next enzyme will be DNA G protein. What will cause the DNA G protein has the tendency to synthesize the RNA primer, because to initiate the DNA replication process there should be a presence of primer. That primer should be 10 to 60 nucleotide long. What happens? The new nucleotides are going to attach in the 3' hydroxyl end, like here. Okay, and as we know, the RNA synthesis, the direction of our RNA DNA replication is 5' to 3' direction. The direction of DNA replication is 5' to 3' direction. So new RNA strands are synthesized in the 5' to 3' direction. Then what happens? DNA polymerase enzyme will attach here. Next enzyme will be DNA polymerase. DNA polymerase. Okay, DNA polymerase have the tendency to synthesize the DNA by joining new nucleotides. It has the property to synthesize DNA by joining new DNA strand, new nucleotides. Okay, there are different types of DNA polymerase enzymes in the prokaryotic cell. In prokaryotes, there is three types of DNA polymerase. Okay, here we will be discussing one by one. DNA polymerase 1, DNA polymerase 2 DNA polymerase 3, and here will be the properties: structural gene, 5' to 3' exonuclease, nucleus activity, 3' to 5' exonuclease activity, this is the proofreading activity. 3' to 5' exonuclease activity means proofreading activity. Okay, the structural gene will be for DNA polymerase will be pol A, for DNA polymerase 2 will be pol B, polymerase A, polymerase B, and here will be the polymerase C. 5' to 3' exonuclease activity present in DNA polymerase 1, yes, will be absent. Here will be present, Further, new nucleotides get attached to the 3' hydroxyl end. There will be need of DNA polymerase 3. This DNA polymerase 3 is a key enzyme for new DNA strand synthesis. Let's see. DNA polymerase 3. It is made up of two units. One is clamp loader and one is core subunit. DNA polymerase 3 made up of two subunits. One is clamp loader, another one is core subunit. Clamp loader has about 6 subunits. They are tied to gamma, delta, delta dash, psi, psi. This helps to load DNA into beta clamp. These subunits helps to DNA. Helps to load DNA into beta clamp. Okay. And core subunit has 3 subunits: alpha subunit, which is actual polymerization synthesis in subunit, actual polymerization subunit. Means it has the capacity, it has the tendency to synthesize new DNA strand. Then will be the epsilon subunit. It has the proofreading activity. Means 3' to 5' exonuclease activity. And next will be theta subunit. Its function not known till now. Function not known till now. Okay, let's draw the structure of DNA polymerase 3 enzyme, how it will look like. Like this. And here will be the okay, this is the gamma complex. It is termed as gamma complex. This is the gamma complex. Means clamp loader. This is the core subunit. Means alpha, epsilon, and theta. Alpha, epsilon, theta. This is the beta clamp. In